Hi, my name is Timofey Prodanov, and I will talk about the work that we did together with Vikas Bansal on variant calling and low copy repeats using short-read whole genome sequencing. Many of you are familiar with segmental duplications. They are also known as low copy repeats, and they are long segments of DNA that appear multiple times across the genome. And by definition, they are longer than 1 kilobase and have sequence similarity over 90%. But we know that in reality they appear much longer and much more similar duplications. And the longer the more similar they are, the harder it is to analyze them. Usually low copy repeats are characterized by a small number of copies in the genome. And these regions are changing for read mapping, not only for short reads, sometimes also for long reads. And as a consequence, they are changing for any downstream analysis, such as variant calling. Additional problem is that these regions are prone to additional copy number variations, and nevertheless, it is crucial to be able to accurately call variants and perform other types of analysis in low copy repeats, because if we consider repeats with similarity over 98%, such regions would contain over 1100 genes and almost 200 of them are disease associated. It's possible that more are disease associated, but these associations are not found yet because these regions are understudied. If we look at the reference genome, repeat copies usually they are not identical, and short differences between these repeat copies are called paralga sequence variants, or PSVs, and they are the main source of information that can be used to differentiate between repeat copies. And consider this example. In the reference genome there is A in the first repeat copy and C in the second repeat copy at the exact same position. And in a diploid genome there would be two A's, two C's. However, it's known that PSVs often coincide with polymorphisms, meaning that instead of the reference allele that would differentiate between repeat copies, we observe allele from the other repeat copy or completely some new allele. And for example, here instead of two A's and two C's, we observe three A's and one C. Or we observe 4 A's and no C's at all, and this is a big problem because now all reads that are mapped to this region, they would map to the first repeat copy. It's completely possible that these alignments would have high mapping quality, especially if there are several nearby unreliable PSVs. One of the most studied duplicated genes is SMN12 because it causes spinal muscular atrophy, a hard childhood disease, and this gene lies completely within 205 kilobase duplication, very long duplication with very high sequence similarity. And if you look at the gene itself, it contains 24 differences, 24 PSVs relative to the second copy. And we also know that only 8 of these 24 PSVs, so small minority, is reliable. All the other PSVs are often identical between the two copies. Additional problem is that for this gene there are common copy number changes across the human population. And in this context, we want to perform variant calling, and variant calling is a very well known problem of finding sequence variants based on existing read alignments. There are multiple existing variant callers such as FreeBayes, JTK, deep variant, and broadly they have two steps. During the first step they discover potential variants, and then for these variants they call genotypes based on read alignments to this variant, and they consider mapping quality, they consider base quality, sometimes they consider nearby variants, but they're not designed for low copy repeats because they don't even have this information about repeated regions, and therefore they don't think that a variant has identical copy somewhere else in the genome, and then analyze these repeat regions independently. Nevertheless, reads and allele observations are dependent. And there are multiple limitations to variant calling to standard method in the low copy repeats. First of all, variant callers often assign low weight or completely ignore reads with low mapping quality. And consider this example. Second copy has heterozygous mutation. It shows AC. First variant is AA. And all reads mapped to the region have low mapping quality because it's a duplication. And instead of calling genotype AAC, variant callers would not call anything. Additionally, if some reads have high mapping quality, some reads have low mapping quality, it can lead to biased variant calls. So suppose reads that have C, for some reason they are assigned low mapping quality, and instead of heterozygous AC on the second copy, variant caller would not call this variant. Even bigger problem is when we have incorrect read mappings with high mapping quality. The problem that can appear when there are unreliable PSVs nearby. Suppose reads that have C, for some reason they are now mapped to the first repeat copy and they have high mapping quality. Then variant callers would call heterozygous AC on the first copy, homozygous AA on the second copy. But the true genotype would be completely the opposite of that. Variant callers do not take copy number variations into consideration, even though these copy number changes are common in duplicated regions. And suppose in this example copy 2 is duplicated additionally one time, so there are 2 C's and 2 A's, and variant callers would probably call just heterozygous AC, even though the true genotype would be ACC on the second copy. Because we are talking about copy number analysis, we already published a targeted tool Parascopy. It uses short read whole genome sequencing for multiple samples, and based on these samples, it estimates distribution of different copy numbers, and it calls aggregate copy number, so some copy number for all repeat copies, and parallel specific copy number when it's possible. And parallel specific copy number is copy number for each repeat copy. And in the process of identifying copy number of a region, Parascopy finds population allele frequencies of PSVs for each PSV and for each repeat copy. And in this example, consider we have 2 copy duplication and we have 2 PSVs. PSV 1 is reliable. On the first copy we will observe A, same as the reference. On the second copy G, also same as the reference. But PSV 2 is unreliable on the second copy. So in the population we always see allele T on the first repeat copy, but on the second repeat copy we observe both alleles T and C, and for that we look at many different samples. Additionally, I would talk about 2 types of genotypes. Parallel specific genotype, where it's the same as the genotypes we are used to, different genotype for each repeat copy. And also we can define an aggregate genotype, basically if we stack all the alleles together. And aggregate genotypes obviously have less information than parallel specific genotypes, but nevertheless they can also provide us some information. And standard variant callers do not call aggregate genotypes. With all of that in mind, we present to you a novel variant caller Parascopy VC. As input it takes short read whole genome sequencing, and also it takes copy number analysis from Parascopy. And here we need both copy number estimates because we want to call variants with different ploidy, and we also use population allele frequencies for the PSVs. And based on all this information, we call variants in low copy repeats, and Parascopy VC finds aggregate genotypes and parallel specific genotypes when it's possible. On the first step, we pull reads from all repeat copies, and this has several benefits. First, this process makes mapping errors and low mapping quality relevant, because even if the read was mapped incorrectly or had low mapping quality, now it's mapped to the same place all the other reads are mapped. And the second benefit is that now we can call variants based on the pulled reads, and we use FreeBayes for variant discovery, and then we call polyploid aggregate genotypes. And once we have aggregate genotypes, and we also have PSV allele frequencies from the population, now we can calculate probabilities of PSVs to have aggregate genotypes that would match the reference. So in this example, PSV1, it is supposed to have TT on the first copy, CC on the second copy, and we look at the aggregate genotype, we see that probably it's true. So this PSV is reliable. However, the second PSV, it has 1C and 3 A's, but we expect it to have 2 C's, 2 A's, so we can say that this PSV is unreliable in this specific sample. And following that, we use paired reads that overlap reliable PSVs and other sequence variants, and we use these observations to place variant alleles on their corresponding repeat copies. So suppose variant 1, it has aggregate genotype AAG, and we observe some reads that go from alternative allele G to the allele T of the PSV1. And using this information, we can place allele G on the blue repeat copy on the bottom. On the first repeat copy, we call heterozygous genotype AG, because we know that G lies at the same repeat copy as T. Similarly, for variant 2, we observe alternative allele T together with PSV allele C, and using this information we can place homozygous TT for variant 2 on the second repeat copy. It's possible that there are no reliable PSVs or no PSVs at all next to some variant, and unfortunately in that case we cannot call parallel specific genotypes. I wanted to add that all of this is done using a probabilistic model, and if there are multiple PSVs nearby, they would increase the signal. And if you want to know more information, more details, please look at our paper. And we benchmark variant calling accuracy across two simulated datasets. The first one where all PSVs are fixed exactly as the reference, and the second where some PSVs are polymorphic, and we took allele frequencies from the European population to do that. Additionally, we looked at 7 high confidence call sets combined by Genome in a Bottle consortium. And because the variant calling method is targeted, we looked at 167 disease-associated duplicated genes, and additionally we confirmed our observations on all low copy repeats from chromosomes 15, 16, and 17. So here are results for simulated data. On the left you can see precision recall curve for the fixed dataset where all PSVs are reliable, and there you can see that all variant callers perform well. I would say that Parascopy yellow and JTK blue perform best of them. But when we go to the right, where we introduce polymorphic PSVs and reliable PSVs, now the results drastically change. Now Parascopy outperforms all the other methods. The second best is deep variant, green line, and dashed line, it shows if we take population allele frequencies for the PSV from a different population, so they would be somewhat incorrect for this specific dataset, we lose a bit of precision recall stays the same. Nevertheless, variant calling accuracy is much better than all the other methods. On the HG002 high confidence call set combined by Genome on the Bottle consortium, I would note that this dataset, it doesn't cover the whole low copy repeat that we looked at. So in more complex regions of these duplications, maybe the consortium couldn't identify all the variants and they removed these regions from the call set. So these results have somewhat less data than for simulated dataset, obviously. Nevertheless, parascopy still performs much better than all the other methods. If we consider the second best deep variant, it has 1% less precision, 5% less recall, and results are similar across all 7 high confidence call sets. And second best is always deep variant. It has comparable precision, so we improve a bit, but we also improve recall by a lot, by over 4%, with also around 4% higher F1 score. And FreeBayes and JTK generally perform worse, and we can show 6 to 8% F1 score improvement. Additionally, we can stratify results based on regions with different sequence similarity, and we can see that at smallest sequence similarity between, for example, 97 and 98%, variant callers usually perform well, and deep variant and and parascopy have relatively the same accuracy. But once we go to very similar regions where sequence similarity is over 99.5%, parascopy massively outperforms other variant callers and have over 20% increase in recall while barely losing any precision at all. Finally, sometimes it's impossible to call parallel-specific genotypes because there are no reliable PSVs. And one example would be CFC1 gene. It is related to cardiovascular diseases, and it lies within a very long duplication with sequence similarity over 99%. And no PSVs are reliable in the population. And nevertheless, even though we cannot call parallel-specific genotypes, we can call aggregate genotypes. And parascopy was able to detect 2 rare heterozygous nonsense mutations across 1,000 genomes cohort, and other variant callers were not able to detect them. So to summarize, we presented a novel variant caller parascopy. We see it finds aggregate variants based on pooled reads, and it uses paired reads to connect reliable PSVs and alleles from other sequence variants in order to place variant alleles for their corresponding repeat copies. We perform multi-locus analysis, meaning that we analyze related regions together, and we use all reads irrespectively of their mapping quality and even if they are incorrectly mapped. The method is multi-threaded and it doesn't require whole genome remapping. And we can achieve higher accuracy than existing variant callers in low copy repeats, and we output aggregate variants that are useful in the absence of other information such as parallel-specific genotypes. And these aggregate variants, they can be used later to associate them with diseases. And you can find variant calling tool and copy number analysis tool on this GitHub. And thank you for listening.